If you look at the volume of therapeutic assets required, today's technology, phosphoramidite chemistry, which is, don't get me wrong, it's fantastic chemistry. It's well advanced, it's refined, but it doesn't scale very well to the volumes that are required to service those potential clinical assets. We're talking metric ton quantities of material required to treat the tens to hundreds of millions of potential patients globally that will benefit from these drugs. And so it is a great time to really think about innovation and new ways of synthesizing these drugs. We are looking at a process that can run under milder conditions in an aqueous environment, thereby eliminating or reducing, at the very least, the need for a lot of toxic chemicals. Organic solvents should be significantly reduced. The need for organic solvents should be significantly reduced.
On the top left, we have a starting oligonucleotide, and we're going to use an enzyme to actually add a nucleotide to it. And our nucleotide here is a 5' triphosphate with a blocking group at the 3' position. So instantly, the eagle-eyed amongst you will realize that we're going to be making our oligonucleotide in a reverse direction to how most of phosphoramidite chemistry does it. It's also phosphorus 5 chemistry. The phosphate's being introduced at the correct oxidation state. So that's our extension cycle. We're then going to need to deblock, and we're going to use another enzyme to do that, to take it off. And I'll talk about the enzymes a little bit later on. The beauty of this approach is that our deblocking enzyme actually is going to deactivate that triphosphate as well. It's going to remove those phosphates, and that's going to deactivate them because the enzyme for extension won't accept them when they're deactivated. And that means we don't have to purify that material away. So that's kind of our core cycle. We're going to go around that cycle to build our oligonucleotide. What do we need our enzymes to do? What do we need our process to do? Well, we need our enzymes to be obviously high performing. And what do I mean by that? So using phosphoramidite parlance, we need to have a high incorporation efficiency here. We need to have a high yield of extension, and we have to have a high yield of the deblocking. The enzyme has to be stable in the process, and those are all things that are visible to the client. But the things that are invisible to you are the enzyme has to be manufacturable. It has to be fermented in a high titer. It needs to be soluble. It needs to be downstream processable, potentially purifiable as well. So those are things that our engineering platform are uniquely poised to do. In terms of the process itself, we are aiming, our key aim here is to be scalable. For this to be a solution, a liquid phase solution approach, so we can leverage high concentrations. We're going to use a liquid phase oligo, and we're going to put our enzyme immobilized. We need to immobilize those enzymes to keep that extending activity away from the deblocking activity. We've yet to prove it, but we hope that the simplified number of unit operations here will actually give us a complementary, and we hope a better impurity profile in the future. And so what I'm going to show you now is, I'm very excited to show it. It's our first ever data with immobilized enzymes doing iterative synthesis. So we're going to show you 6 extensions here. The oligo that we're starting with at the top in teal is FAM labeled, so this is a CE trace here. We've got a linker, then we end in a modified ribonucleotide. And so the nucleotides that we've added here are in the darker color, methoxy A, fluoro G, methoxy U, etc. And you can see as we go along down to the 6th extension, we're getting a good reaction. We can see some material that's still left here. We fermented this enzyme 2 months ago and have been doing this work on it. So, our enzyme now has even better capabilities than this, but this is the data that we can share at the moment. So we're super pumped about this. Like I say, this is the first time that anyone has actually done this with a non-template dependent polymerase with solution phase oligonucleotide. And we're continuing to engineer this enzyme now. I think I will point out the eagle-eyed, you can see that extension number 5 and number 6, they seem very close together. We did have an authentic standard that material made, and we got mass spec on that. So it's just an unusual way that it runs on the CE, but it is the desired product. So, like I said, to our knowledge, this is the first example of this using immobilized enzymes. We showed you 6 extensions there. And this was all done, very small scale in the lab, in essentially like an Eppendorf, transferring solutions in batch mode. And where we're moving to now is we're going to move into more like a packed bed mode, and we're going to start moving solutions and circulating solutions across these immobilized enzymes to make it more process relevant and into like an automatable form factor in the future.
Our immediate future goal is to develop this technology so that it can perform on par, or I firmly believe can actually exceed the performance of phosphoramidite chemistry processes in place today. With that technology developed, I think the vision for a greener, potentially more sustainable process for manufacturing these important drug molecules is very much a reality, an achievable reality. In addition to the improvements on the environment, there's also a real opportunity to improve the quality and the quantity of the API that is being manufactured by these enzymatic solutions. So all combined, I think there is an immediate benefit or long-term benefit that we'd like to see for patients.